Hi, I'm Tim Colmego from ImageIQ. I'm here today with Dr. Amit Vasanji, our Chief Technology and Science Officer. Happy July. Yeah. Happy August, actually. It's August already. I can't believe it. Holy cow. We are here today to talk a little bit about some of the work we're doing in preclinical imaging analysis. And this series of mini webinars is produced by ImageIQ to discuss kind of the advantages and disadvantages of analysis of different kinds of preclinical imaging that you might be doing. Take advantage of your expertise in doing this for probably more years than you care to admit at this point. Right. It seems like just yesterday you started, right? Today's webinar is a little bit different. Normally we kind of have a nice rhythm where we talk about the types of researchers and talk about the field of research and then the pitfalls of the imaging, and then we talk about the bot, and we're done in kind of like 5 minutes. This one's probably going to go a little bit longer because we're really going to talk about 3 similar assays that involve quantifying neural images, right? Right. So we're going to talk about cortical neuron and astrocyte analysis. Is that fair? So perfect. So let's start with the research. I mean, obviously, given that it's cortical neurons and stuff like that, it sounds pretty obvious. But who's doing this kind of research? What kind of stuff are they looking at?
Essentially, it's people that are researchers that are involved in neurobiology. So anyone that's looking at cells that are coming from the brain or the spinal cord, they're doing this sort of in vitro work and analyzing these cells. So.
Great. And with that kind of stuff, I mean, it's probably all the popular neuro kind of research, people looking at MS and Alzheimer's.
Right.
I know like right now we're talking about traumatic brain injury and that stuff's kind of at front and center. So that's kind of where it works. What kind of are the goals of the assay? What are they looking for when they're doing this?
They're essentially identifying extensions and other staining patterns that are in these neurons. So as long as you have a base marker that identifies all of them, the astrocytes of the neurons, then you can look at other markers within these cells and then look at extensions and if these markers are within the extensions or not, or the neurites or processes that are coming out of them.
Okay, so let's talk a little bit about then the image acquisition, because we're really talking about a couple, 3 different kinds of ways. What's the process for them? How's the image acquired? What kind of happens there?
So it's just a fluorescent staining assay, so you're labeling it with a marker that's known for those particular cells. So for astrocytes, the common marker is GFAP, which are filament markers. So they will label all the filaments in all of these astrocytes. For neurons, the most common marker is tubulin, which is another structural protein within all of these neurons. So as long as you stain with those, and they usually use a DAPI, which is a nuclear marker, to identify where all of the soma are within these cells. You're using a conventional microscope to take these images. The hardest part is actually isolating and prepping these. One of the things that you probably want to consider when you're doing this is not to wash them too much. So these neurons are forming these neurites that are really they're not necessarily stuck down to the bottom of the dish. And they can sometimes float around, and what you don't want to do is wash them so hard that the neurons start curling around the soma or touching other neurons. So it's going to be hard to sort of
So you kind of want to leave them intact and not
prepping them is one of the most important things.
Got it. And is that probably the most common pitfall? Since it's a standard, like you said, fluorescent kind of analysis, I mean, it's really... Right. There's no super protocols that are going on from a microscope perspective.
It's not really. I mean, we have 2 bots on purpose for low and high density astrocytes because it's so hard to tease them apart from one another. But for the cortical neurons, if you're interested in analyzing each individual cortical neuron, make sure they're sparsely sort of seated onto a dish. So.
Got it. Well, I guess that brings us to kind of the next step, right? Which is kind of what's the standard of analysis. If you look at this image that's on the screen right now, for those of you who are watching, we're looking at the screen up here. Technically, I think it's like over there. You got to do like the weatherman thing, right? But if you look at that image, it's pretty complicated, right? There's a lot of data to try to get out of that. How does anybody even try to do this manually? What happens?
You can try. There's always a way to try to get these different staining patterns and extensions and processes analyzed, but there's so many, and it's hard to identify which one belongs to which cell. So, and you've got thousands of cells in this field. So, depending on how many stains and markers you're using and how dense these cells are, it could take hours per field. So,
and I imagine there's a ton of bias that gets introduced. I mean, we see it when people get tired and they get whatever that gets kind of crazy, but. If you're trying to use off-the-shelf software, right, and we talk about this a lot when we start talking about the IQ bots, and that's coming shortly, but when you use off-the-shelf software, you've got to make sure that you're doing something that is controlled and consistent, right, and works within your budget and all that kind of stuff. And we see people who struggle with this, especially when the images get complex like this, where they're trying to use some piece of software and they just can't. Once they get it to work, then they try to get it to work on their next batch of data or next batch of images, and they can't even reproduce the results and all that kind of stuff. So what's out there? What do people try to do?
There's a number of software out there. You can use MATLAB or ImageJ or Cell Profiler. You still have to put in rules for how each of those are analyzed. And if you're most likely you don't have an image analysis expert in your lab. It's somebody that's been sort of co-opted to do the actual work, and they may not understand the tools and how you can change them or to make them robust enough to run across all of those variations you just talked about.
Got it. Well, I guess that brings us to kind of why we like the image quantify concept and our concept of IQ bots.
Right.
We at ImageIQ, we created something. We saw these challenges, and we realized that people were really struggling with that. And to put it very simply, I think the best way to say it is, our clients and customers of Image Quantify just want to do their science. They want to be good bioscientists, biomedical researchers, that kind of stuff. And they're not software folks, and they're really not all of them are imaging folks, right? So we created this thing called IQ bots that live on imagequantify.com, and the basic idea is you upload your images to the website, you select the right bot. So in this case, we're talking about 3, right? The cortical neuron ones, and then the high density and low density, right? And you select the right bot for the kind of image you have, and then the imagequantify.com site will actually process that bot and give you a bunch of output data, right? And strangely enough, when you're doing these kinds of things, we like it because it's we think it's better, and it's better science, and it's more cost effective, and it's easier for the folks in the lab. They get their results kind of right away. They don't have to struggle with this thing for a week to get the analysis working. But when we show images about this, we always like to talk about the spreadsheets, but we never put the spreadsheets on the screen, right? Because the images look cool.
And the spreadsheet won't fit. So.
Right, yeah, so they're pretty big. So when you're doing, let's start maybe with the astrocyte analysis, take a look at a couple of those images and talk about, okay, what kind of output metrics do we get from that?
Right, so there are 2 flavors for this particular assay. So we have a low density and a high density. So the low density is on the left here, and they're pretty well spread out. The common marker again is the GFAP. So the IQ bot identifies each of the individual astrocytes for the low density, and then you can choose to either analyze them all, whether or not they're connected, but it actually looks to see if these astrocytes are connected, and this particular bot only looks at the ones that are isolated. So,
got it. Okay.
And then it identifies CSPG, which is in this case another antibody that you're within these cells, and it does it in 2 different ways. It looks to see if the CSPG is present and how intense it is, and then the total area of the puncta that it forms, because it doesn't necessarily form a uniform stain. It actually forms different sort of different clusters of these.
Which is another reason why doing any kind of manual analysis would be
correct. For the one on the right, these are super dense, so it's kind of hard to identify which of these the stains belong to which nucleus. So what we did was actually create these sort of cages around each of the nuclei. So identify each nuclei, and then the mean distance between each nucleus, it finds a bisector and it separates them out. And so you end up forming these cages around each nucleus, and then within that cage we analyze how much GFAP there is and how much CSPG there is in there. So.
Got it. That's super cool. And then all that data gets put into a big spreadsheet, right?
Right.
That we're not showing here.
Right. And then you get these overlays as well. So for each image analyzed, you get an overlay and you get the spreadsheet. And this particular bot in the cortical neuron one was validated across, I think, thousands of images, literally, with a particular colleague that we were working with.
Got it. Very cool. So let's talk a little bit about the cortical neuron one, right? So how does that one work? It's a little different kind of imagery.
Right. So in this case, the main marker for identifying these particular cells is tubulin. So tubulin is a structural protein, and it labels all of the processes, and the soma, which is the cell body of the neuron. So the algorithm finds each of these neurons, and then it determines whether they're connected or not, because it's really hard to judge whether a process which process it belongs to if there's 2 cells or 3 cells connected to one another. So it identifies only the neurons that are separate from one another in the actual field. And then for that identified neuron, it finds the cell body, or the soma. It finds the mean intensity of the soma, it finds the area of the soma, and then it looks at each of the different extensions that are coming out of the soma called neurites, right? Okay. And there could be anywhere from 2 or 3 to like 30, 40.
Yeah, I remember the team doing the validation on some of this stuff here in-house, aside from the partner stuff, and it was like tracing those things and drawing them, everyone was kind
Right.
of just like running out of the building screaming.
Right. And so, talking about the big Excel sheet, for each neuron that it analyzes, you get a count for each of the neurites that are coming out of it, and then a length of each of the neurites that are coming out of it, and the mean thickness, the mean intensity of the neurite, and then it finds the longest one, and it finds the number of branches that are coming off the longest one. So,
And that's relevant usually for this kind of research?
Yeah, so in some cases that longest one may be an axon, so they're identifying that as the longest neurite.
Got it.
So.
Got it. Super cool. And that all goes in a big spreadsheet, and then
Correct.
You hand that over to your biostats people. Their head pops off.
Right.
And they're
Right.
like, oh my gosh, what do we do? And like we've said in the past, you can take that raw data and then start doing something different with it. Looking at histograms, looking at binning, however you want to. Do something with it, because it is a lot of data.
Right. And if you had a particular set of these images and you wanted to know, is my marker in all of the processes that are coming out, you just create another channel, and then we just input it, and then for each identified process, it finds the intensity of your marker.
Got it. Perfect. Well, that's great. Well, hey, everyone, thanks for watching again. I mean, thanks for the time and sharing, for sure. We love doing these things. We love sharing with you, kind of the experience that we all have with imaging and doing some really good stuff. If you have any other additional questions or anything, you're welcome to visit us at imagequantify.com. You can send us an email. You can see our phone number up there if you want to just give us a call, whatever works. But again, thanks for watching. We're looking forward to making many more of these. If you have any feedback, let us know. We're happy to try to make these more and more useful for everybody, because that's really our goal in life. So, thanks very much.